Okay, hello, good evening. Welcome to week 12. So today we will begin our discussion with a quick recap of the last class, followed by a discussion on sequence alignment tools. Mainly, I'll be discussing the principle and the mechanism that is followed by a very common known aligner, which is the star aligner. Of course, there are other examples as well, different sorts of protocols or standardized pipelines. They follow different, or they preferably use certain types of tools, but STAR is a very common one, and we'll be discussing that for the day. Following that, I will be quickly revisiting the protocol not the protocol, I will be quickly revisiting the topic we have already covered in the first couple of weeks. So we had discussed gene structure, function regulation, mutations, polymorphisms, and functional genomics, introduction to sequencing methods, and unraveling the genomicsome and transcriptome, and also introduction to epigenetics and understanding data methylation and histone modifications. These four or five topics are the ones that I will be discussing. Um, just quickly brushing up today so that we can have a small test in the next week, following which we will shift gears and move on to our discussion um, regarding strategies to map DNA methylation that can be used to detect the genome. And then we'll move on to DNA protein interactions, the techniques that are used commonly in the lab, and how you can convert them to high throughput methods, and so on and so forth. We'll take it onwards from that point. So today we'll keep our new discussion a little brief, and we'll mainly stick to revision of these older topics so that it's fresh in everyone's minds before our discussion for the next week. So, if you remember, in the last week we had started our discussion with introduction to the reference genome. So, the reference genome, the topic or the idea by itself was brought forth and was it was actually the accomplishment which came out of a huge project, a huge initiative called as the Human Genome Project, or the HGP, which was an international scientific research project that had the idea to basically unravel the sequence of the entire human genome. And what does that include? That includes collecting DNA from multiple different sorts of populations from different sites, from different hospitals, which were collaborating as part of the project, and then decoding the different regions of the genome using the strategies we have already discussed in previous classes. And overall, the idea was to have an idea of what all which portions of the genome are not very known to us. Secondly, the idea was to look for biomarkers to identify variants to carry out population-based studies, and of course to eventually benefit mankind. So this snapshot overall shows you the progress that took place as part of global research that eventually led to the onset and the completion of the Human Genome Project. So it began in somewhere in the 90s, early 90s, where you had a lot of hospitals collaborate for the same. You had a couple of genome centers that were established majorly in the United States, following which, following some guidelines, the 5-year plan was set up somewhere around 1993. The goal was achieved in 1994. Then, if you have some background in genetics, you would know that the complete genome sequencing of a number of different other organisms was completed before the Human Genome Project. That includes C. elegans and a few others. Obviously, organisms whose genome was a lot less complex were sequenced before. And yeah, all of these eventually contributed to the same. So yeah, somewhere around here, C. elegans genome was sequenced. Yeast was actually the first eukaryote whose genome was sequenced. Then you have E. coli genome sequencing, roundworm or C. elegans genome sequencing, where the SNP initiative began, which is the single nucleotide polymorphism initiative. It began. Then human chromosome number 22 was the first human chromosome to be sequenced. And then you can see the citation of a number of different publications, which altogether have eventually led us to where we are today. So this was a snapshot of all of the different institutes which were collaborating as part of this project. Like as you can see here, most of the centers or the or the collaborating institutions were part of the United States, but there were representations from United Kingdom, Japan, France, Germany, etc. as well. So that basically contributes to the diversity of the input genome or the input DNA sequence, which was used for sequencing, and therefore the consolidated output of all of these independent individual sequencing projects can altogether mount to a sort of a representative of all of these consensus sequences that can be used as a reference across different populations. Um, but despite the initiative and the magnitude at which it was carried out, one of the major shortcomings was with the one of the major shortcomings that were encountered was the fact that majorly only the euchromatic regions of the nuclear genome were sequenced or could be sequenced, which left out a good portion of around 80%, which was somewhere scattered in heterochromatin regions. And that has been modified over time. A lot of these sequences have already been decoded. But of course, there's improvement or there's scope for improvement that's still somewhere around. So then in March 2009, okay, while the first initial draft was available in June 2000, and in 2001, the working draft was completed and published in 2003. There were further progress was made, and in March 2009, the Genome Research Reference Consortium released a more sort of accurate version of the human genome that left more than 300 gaps. But yeah, that could be completed. Then in May 2020, the GRC reported 79 unresolved gaps, accounting for as much as 5% of the human genome months later. And in April 2022, the Telomere to Telomere Consortium, the T2T Consortium, published a complete set of the non-Y chromosomes. And in August 2023, Ree et al. Reported successful complete sequencing of whatever remaining portion stayed back from the Y chromosome, finally achieving full sequencing of all 24 human chromosomes. Oh, sorry. Yeah. Then the idea that we discussed next was that how is the sequencing or how was the sequencing initiative even carried out? So basically, a special sort of a vector was employed for this purpose, which was BAC or bacterial artificial chromosomes, which have been whose job or whose um, key attribute lies in the fact that usually you'd be able to clone very large sequences into these vectors. They are able to make large inserts, and B, they are capable of stably being transformed across bacterial generations, which allows amplification of DNA. Now, you need to keep in mind that this was a strategy that was used back in the day. There has been progress made in this field as well, but of course, we'll start with where projects were initially where they were initially beginning and what progress has been made over time. So, majorly the key advantage lies in the fact that you can use these back vectors and you can clone up to 300, 400 base pairs of DNA, and it works perfectly like any other vector plasmid, where it would have an independent origin of replication, resistance genes, which allow selection, drug-based selection, and you can have something similar to a multiple cloning site. It is not as small or concise or not as convenient as that of a regular plasmid, but certainly very, very useful. So yeah, so you clone your sequence of interest, which in this case, let us call it the GOI, which you cut using a certain set of restriction enzymes, which should be compatible with the vector as well. Now, the bacterial F prime plasmid is cut with the lactase gene with restriction enzymes, and then cloned, and the completed or covalently closed insert can then be electroporated into E. coli cells, and then they would be plated onto X-gal IPTG medium. Yeah. So then they can be plated onto X-gal IPTG medium, and then after transformation, based on blue-white screening, you'd be able to select for those populations that successfully have been transformed and then those bacterial populations would be grown, and more and more amplification of your target sequence would be feasible under those circumstances, and then they can be used for DNA isolation and for sequencing. So it's just a strategy for high level amplification of your gene sequence of interest or the insert of your interest using bacterial artificial chromosomes. Now, with respect to the reference genome, these were some of the key institutes that participated into this project, and they formed the GRC, or the Genome Reference Consortium. The Wellcome Sanger Institute, the McDonald Genome Institute, European Bioinformatics Institutes, NCBI, Zebrafish Model Organism Database, Flat Genome Database, etc. Now, how you can actually access this information. Now, keep in mind that this keeps on getting updated every couple of years because, as we've already discussed, in the last approximately 2 decades, a lot of progress has been made, and every time there is an update that is being rolled out, you can access it using these publicly available portals. Like, for example, the UCSC Genome Browser, very commonly used, is a wonderful tool for you to utilize publicly available data sets. And if you go into details about the current Human Genome Browser, you would see that the assembly is HG38. The previous one was HG19, and it was part of this particular project. And you can see there's a lot of other information also available. Now, why this information, particularly HG38, is necessary is because when you are carrying out data analysis and you want to make sense of whatever output you've gotten, then the tools that you use sometimes can have different compatibility. What do I mean by compatibility? They might be compatible with HG19 or HG38, depending on whether an update of that tool has been rolled out. So whenever you are carrying out SIM analysis, make sure you check this compatibility and accordingly select the tool that you want to proceed with. So this was the information about the reference genome. So next, the question that we address is how do we make sense of all of this information? So for that, if you remember where we had left off was that okay, just you were not present in the last class, so I'll just give you a quick recap of what these topics have already been discussed in the last class. I'm quickly recapping week 11 so that I can move on to today's discussion, which also will keep a little brief. We'll only be discussing star aligner and alignment tools for today, which will cover most of the analysis-based tools that I wanted to discuss. So what we're going to do after that is do a recap of the topics we have covered initially, majorly centered around molecular biology, because as I mentioned in the previous class, we'll be having a quick MCQ in the next week. I will drop an email as to when that can be done, probably Tuesday itself, but I'll drop an email about it. So you can just brush up those topics. Nothing too difficult, nothing too serious. But yeah, certainly we want to keep those topics relevant. Okay, that is one thing. Now coming back here. Yeah, so we were discussing the principle of alignment. So if you remember, we had first sonicated our sequences, shotgun sequencing was performed for that. And then the reads that you get, we were trying to make sense of those reads. For that, first we did pre-processing of our data using tools like FastQ. Then we trimmed our data to get rid of adapters or any other non-specific sequences which are going to mess uh, with the remaining analysis portion. And then we moved on to the next step, which was alignment. Now, when we talk about alignment, we first addressed what is the concept of a reference genome, because that is the template to which you'll be aligning your reads. So continuing with that discussion, we're going to take it forward. So if you look here, you'll see that these are different sources from which you can actually get the human genome sequence and the annotation files, the GenCode, UCSC Genome Browser, Ensemble, NCBI, RefSec, etc. I would recommend you to go for GenCode. You'll get the most recent rollout, and that is what you should always take into consideration whenever you are proceeding with alignment. And if you have performed analysis say today, and then a year from now you expect to do the analysis for the same set of samples, then it's fine to use the older version. But if you have a different sample, or if there's some incompatibility between the two, it's always advisable to have the same reference genome. So you would have to reanalyze your data 1 year from now if you wish to include them as part of the same experiment. So all of these little details need to be taken care of, because otherwise they're going to skew your results, and of course that's not something you want. So yeah, now, what do we do practically as part of our analysis? So, once you have your genome sequence from these resources and you have your annotation file, you have to index your genome, and that is usually created by the software you're using for alignment. Now, the one I'll be discussing today is Star Aligner. So, we'll come to more details about it later on, but these are some of the most common tools: Bowtie 2, Star Aligner. You also have other tools like TopHat, you have HiSat2, etc. These are all different tools which have some preference in one type of technique, but technically speaking, they are all alignment tools. Their principles might be different, or the algorithm they follow might be different. And some standard pipelines use some specific tools. So that is something you should certainly refer to. Maybe on GitHub, maybe go through their manuals before you proceed with the analysis. Okay, now now we're talking about generating genome index. So, why at all are we coming to this objective? Why are we talking about indexing at all? So, one of the major problems with this immense amount of information is that it requires a lot of computational power to scan through the entire genome for each read, to carry out this scanning and then assign it its position and then come to a conclusion about this. So similarly, star algorithm also requires around 30 GB memory free to align human or mouse genomes. And therefore, it is an intensive process. So you can refer to this particular manual for details about how the star algorithm actually works. It's a beautiful manual if you are somebody who is actively, doing any sort of analysis at this moment. You'll get a lot of help here as to what code you should use if you're working with Linux systems. So yeah, now we were talking about generating the genome index using star. So this is what the example code basically looks like. This is something you just have to adjust based on what where your files are stored, and then you run this code, and that'll basically index the genome. So the question is, why are we so bothered about this process of indexing? And what is the problem with sequence alignment that we're talking about in so much detail? So first of all, understand this fact: this particular step is very crucial. Till now, whatever you have done is simply cleaning up your data and making it ready for analysis. Now, if you are talking about ready for analysis, then where exactly does the analysis part start? It's a sort of chicken and egg sort of an example, where these pre-processing steps and data cleaning steps are also part of analysis, but the major step lies in exactly aligning your read, your output sequence, to a template. So this step is extremely crucial. And what is another factor that needs to be taken into consideration is that time, the amount of time and the amount of computational power which you dedicate to this particular step should also be adjusted to make it most feasible. So, like I previously mentioned, this is the major bottleneck in this process, whereby finding the correct position in the reference genome from where that one single read has come is a very challenging process. Why is it especially more challenging? Because there are certain regions of the genome which are quite repetitive, and because your read length can usually range somewhere around 50 to 150 base pairs. On an average, it is around 100 base pairs. So as a result, what happens? It might find homology in a large number of, not homology, complementarity in a large number of regions which are quite repetitive, and then that can also affect your results. So this is one more bottleneck. And third, a certain degree of flexibility for differences to the reference genome should also be allowed. It cannot be extremely stringent that you want 100% match between your read and your reference genome. Why? Because it's possible that there are point mutations and there are some variants, and your objective is to actually find those variants, right? So if you put too stringent a criteria, then it is possible that it doesn't align anywhere, and then you are losing out on data. So this is where indexing, the process we were discussing, comes in handy. So all alignment algorithms, they use some data structures which are called as indices that allow you to, sort of sort the data. Think of it like breaking down a large puzzle, like yeah, a large object into smaller objects, into smaller cubes, and then individually looking at those cubes using multiple threads at a time so that your alignment time is cut down by a lot and you do a much faster processing of an immense amount of data. So, with the help of these algorithms, it's possible to compare over 100 gigabytes of sequence data from NGS analysis with human reference genome in only a couple of hours. So, usually, for example, like for RNA-seq, if you have a 100 base pair sequencing which has been done, and you get around 3 or 3.5 GB data, if your processor is good enough, star aligner only takes like 4 to 5 hours, which is immense considering the amount of genome it is actually searching. It can take even less depending upon how powerful your system is. But I just wanted to give you a realistic sort of estimate. So yeah, so sequence alignment is this process of arranging these DNA RNA sequences and two regions of similarity. And an optimal alignment is one that has minimum number of errors, where it has minimum deletions, insertions, or mismatches. And if there is no error, then that is an exact match. So this is a representation of what alignment pretty much looks like. So when you have all your puzzle pieces aligned, it gives you a logical output, an actual image. And so that is the conclusion. So then we talked about two algorithms which are commonly used. So sequence alignment is used in molecular biology. Um, Two types of alignments are possible under these contexts. One is global alignment, the other is local alignment. We watched some videos also last week to understand how exactly the scoring for these different algorithms is done. I'll not go into details of that today. I'll upload the videos, so if you want, you can refer to the recordings for the class. But majorly, the idea is that you want to, if your alignment takes a large sequence at a time, if that is the case, then it's a global form of alignment. If it is something that is taking shorter regions or small fragments and into consideration for your alignment, and where there is no negative scoring, then that is a local alignment. The algorithms are different. It's called Smith-Waterman, and the first one is called Needleman-Wunsch. So that was pretty much the idea we discussed in the last class. So taking this forward in today's class, we'll be just quickly understanding star aligner, its principle, what it does, and how it does it, and then we'll move on to the recap that I was talking about a while ago. So, if you remember, this was just to quick recap the steps we have covered. So, first is your library preparation. First, you have your biological samples from which you isolate DNA, RNA, whatever is your starting material. Then you prepare a library out of it, which undergoes amplification and enriches your sequences at a level which makes screening easier. Um, and out of that, when you subject those sequences, when you allow sequencing to be completed, the reads that you get are your output files, which will be represented as .fastq files, which will then undergo pre-processing in the form of fastqc. Already we have discussed the many different parameters which come out of it, which tell you about the quality of your data, which tell you whether or not any sort of edits are needed in the same, whether you need to cut down any sequences, eliminate any sequences, etc. And along with that, you also can do adaptive trimming, which, while mentioned here as optional, is always recommended so that it doesn't, mess with your actual sequences of interest. Then you have genome mapping, reference genome mapping. So here you see a term splice aware. I'll explain it in a little while. And then you check how many reads out of your mapping, how many of those reads are associated with your genes of interest so that you can draw reliable conclusions about which genes have undergone changes between your control and your experimental sample. So, and then you do statistical analysis. You check whether there are whether there are any differentially expressed genes or DEGs, or in some cases, when you are using whole exome data, you might be interested in finding mutations, in finding some single nucleotide polymorphisms, etc. So, depending on your objective, you do the downstream analysis. So, that part we'll cover towards the end of the course. But for now, we are going to somewhere take a pause somewhere here, and then we can take it on later once we have discussed other sort of analyses. So, to basically determine where on the human genome our reads have originated from, we can align our reads to the reference genome using STAR. STAR stands for Splice Transcripts Alignment to our reference. It's an aligner that is specifically designed to address the challenges of RNA-seq data mapping using a strategy to account for spliced alignments. So it basically allows you to search the genome and find sequences to which your sequence of interest is able to align. How does it do this? It's a standalone software that uses sequential maximum mappable seed search, followed by seed clustering and stitching. Don't worry about it. We'll look at each of these steps in the coming few slides. To align RNA-seq reads, what is its unique feature, or what are its useful attributes? First, it is able to detect canonical junctions, non-canonical splicing, and the presence of chimeric transcripts. So why knowing this is important is because you would then be able to, you know, understand in depth why certain tools are better suited for certain types of purposes. So Star Aligner, it is one of its major commendable features is that it has very high accuracy and it actually outperforms other aligning tools by a factor of more than 50 in mapping speed. But its shortcoming lies in the fact that it is a memory intensive tool, and the algorithm achieves this highly efficient mapping by performing a two-step process. What are those two-step processes? It does two things. First, it does something called as seed searching, and two, it does clustering, stitching, and scoring. So we'll come to this in the next slide. So keep in mind, this is the job of the tool. You do not get to see any of these steps. It's just something that's happening while your program is running in the background. This is how the tool is doing its job. So for every read that the star algorithm or the star aligner is able to align, what will it do first? It will first search for the longest sequence that matches exactly one or more locations on the reference genome. So this is your read sequence. Again, all of these information I have taken from one of two sources, either from the STAR manual or I've taken it from GitHub. For same, again, the official site for STAR manual or the STAR aligner tool. Again, keep in mind, I have not included every possible information. There's more and more depth you can go to. But just so that you know the principle, just so you know the logic and the biology of it, it's not even biology, but the logic of it. I've included some portions. You can learn about all of this in as much detail as per your interest whenever you have time. So like I was saying, what this tool basically does is that it searches for the longest sequence that exactly matches one or more locations on the reference genome. So this is your read. Consider this to be a 100 base pair long sequence. Now, when STAR is trying to find regions of complementarity, regions where they actually match, not complementarity, regions of homology, or regions of matching between the read and the reference genome, it might so happen that it does not find the complete sequence. All of the 100 base pairs may not be exactly aligning somewhere in the reference genome. Instead, what it might see is that there is a portion which is matching exactly with the reference, and there is some portion which is an unmapped portion of the read. The different parts of this read that are separately mapped can be called as seeds. And the longest matching sequence is called as an MMP, or mappable prefixes. So this part is an MMP, and these separate individually mapped parts would be called as seeds. So, like we were initially discussing, that seed searching is the first thing that Star Aligner as a tool does. It basically looks for longest regions of your reads that can align with some portion of the reference genome. Please let me know if you do not understand at any point. We'll go through it again. So the first MMP that gets mapped to the genome is called as seed 1. Star will then search again for only the unmapped portion of the read to find the next longest sequence that exactly matches the reference genome, which will be called as seed 2. So it pretty much does the same thing, but for the unmapped portion of the genome. So like you can see for this seed 2, it has found a separate site. And what is the most logical conclusion then? That this particular read has come from a donor and an acceptor, which are actually far apart on the reference genome, but have eventually resulted in one common read. Why is this necessary? This is necessary because Star Aligner is a tool which is used for analysis of RNA sequencing data. So what will you, in those circumstances, what will you expect? You expect that these were part of some overlap. Yeah. So what would you expect in those circumstances are that these two have actually been overlapping a splice junction, and as a result, when you have one read which corresponds to two different far apart sequences or sequences which are apart on the reference genome, they have come from a splice site. That is why we called Star Aligner as a splice aware aligner, which is capable of detecting such splice junctions. If it was not a splice aware aligner, what would we expect in those circumstances? Or just consider any hypothetical tool that is not able to detect such junctions, and in those circumstances, if it does not find sufficient match of the read with any portion of the coding sequence, it might just drop that read. So what will happen? You will end up losing out on data, and incomplete information would be brought forth, and therefore such tools are not recommended or not usable for RNA-seq data analysis. So taking this into consideration, Star Aligner is a good tool and a very commonly relied upon tool as well when you are doing RNA-seq data analysis. So this sequential searching of only the unmapped portion of the reads underlies the efficiency of Star algorithm. Star uses an uncompressed suffix array to efficiently search for these maximal portions, which are the longest sequences that are finding an alignment with the reference genome, which allows for quick searching against even the largest reference genome. Other slower aligners, they use algorithms that often search the entire read sequence before splitting reads and then perform iterative rounds of mapping. So as you can understand, that requires A, more time, B, can incorporate more inaccuracies, and C is going to cost you unnecessary computational power. So in these aspects, Star Aligner is a lot smarter, a lot more efficient, and is going to do the job in a much shorter time. If Star does not find an exact matching sequence for each part of a read, either due to some mismatch or due to some indels, then what happens is that the previous MMP will be extended, and that will be the sequence that will be taken into consideration. So here's what you see in this context. You have this read of which, for example, this is your MMP. This is a region which has found alignment with the donor site. However, your unmapped portion of the read, which was C2, is carrying some sort of a mismatch or some sort of a variation from the reference genome. Uh, so in case it is able to find or is able to incorporate this mismatch, then Star Aligner will be able to proceed ahead. But if it does not find the exact matching sequence due to these mismatches, then the previous MMP gets extended, and this portion, basically this MMP, this will get extended, and that entire region will be considered to be part of a gene which will be used for further analysis. In case extension is not giving, good enough alignment, then poor quality or adapter sequence or other contaminating sequences can be soft clipped. So this is where soft clipping would be done, and only this much portion of the read will be used for alignment, and then also it can be used for further analysis and for comparisons, taking into consideration that there is some sort of a splice site in between these two exons. So basically, this was how the tool exactly works. And now the information that you get from these individual seeds getting aligned will have to be stitched together so that a complete read or can be, constructed. So you have these two reads or these two seed sequences aligning to these regions of the reference genome. And when you stitch them, you will get the complete information. So what it does is it first clusters the seeds together based on proximity to a set of anchor seeds. The seeds are then stitched together based on the best alignment for the read. Again, these are all processes or steps which are occurring in the back end. You don't have to actively do any of these things. But if you want, you can tweak certain processes based on your experimental goals. And yeah, that is pretty much how Star Aligner works. Apart from that, there are many other tools whose names also I previously mentioned a little briefly, but you majorly have Bowtie and Bowtie 2. So Bowtie is an ultra-fast, memory-efficient, short-read alignment tool which is used for large reference genomes, and it has been indexed with Burroughs-Wheeler index. Typically, its job is to align DNA sequences because it's a splice-unaware tool. Keep in mind, this is why I mentioned that understanding why splice-aware tools are relevant and why they are preferably obviously used for RNA-seq data analysis. But for DNA sequencing data analysis, you can use Bowtie. Apart from Bowtie, there's also Bowtie 2, which is a different version of that software, and Bowtie 2 is actually more commonly used. These are also ultra-fast, memory-efficient tools, which are suitable for aligning reads around 50 sometimes even goes up to hundreds or thousands of characters to very long reference uh, sequences. So, yeah, these are some of the common tools and what you actually expect as output of this alignment. So, you let star algorithm do its job, you give it whatever amount of time to do its job, say 4 to 5 hours, and after that, what you'll get as the output is this file format, which we have already discussed in our, I think, very first class when we started data analysis. So SAM would be the output file that you would get, .sam, which stands for sequence alignment or map format. And what is something you do immediately after getting this output? Again, this is something we discussed previously. You basically convert the SAM file into a BAM file. So same data, just the format has changed. And as you can see from this image as well, SAM is a text-based format. It's a tab delimited text format, and BAM is a binary format, and it's a much more compressed version of the SAM file that is used for representation of aligned sequences. And all further analysis steps, whatever is suited to your experimental um, goals, will be carried out using the BAM file. And in the BAM file, it contains a header section and alignment section. Header contains information about the entire file, sample name, etc., what method has been used for alignment, and under alignment section, you have the name of the read, its sequence, the quality, alignment information, and other custom tags. So yeah, that is all the information that is sort of fit into a BAM file, and that makes the file much smaller and more usable for further analysis. So I think yeah, that is it for today's topic. Next, we'll start our revision for the initial discussions. If you have anything in mind that you wish to clarify till now about these data analysis steps, please let me know, and then we can begin discussion or revision for week one, from week one onwards. Just unmute yourself and you can let me know. Okay.
Ma'am,
Yeah, go ahead.
ma'am, can we consider mitochondrial genomics, genome sequencing when we do our genome sequencing of our genetic material, which is our DNA, along with our
Uh,
mitochondrial genome?
No, so you will get rid of that information as well, right? It
Hmm.
actually is something that you select during the library preparation step. So, for example, let us assume you're shipping your samples to some company, your cells or tissue, whatever it is, you will be able to select the library which you want to use for sequencing. So if you want mitochondrial information, then you can select that, and they'll give you a like. If you can actually go through the Illumina website, they'll give you an idea of what all possible libraries exist. So if you want, they will enrich for mitochondrial DNA specifically, but usually people, unless they are working with mitochondrial genome, do not want it. So it is not the most default protocol. Um, but what you often do get is microbial genome. The If you have sent a tissue sample, you will often get the associated microbiota that is associated with the by default with the tissue sample. That information is often gotten along with your sequence of interest, so that needs to be subtracted from your actual DNA and actual RNA so that it's not a contaminant. And that is done using, again, using tools. You cannot do it any other way. But if you are interested in studying the mitochondrial genome, you can do that.
Thank you, ma'am.
Okay. Yeah, just to, because you joined later, Vaishnavi, just to let you know, I had discussed the exam in the last class, but I'm very busy this week. It's not going to be possible. We'll have the quick test next Tuesday itself in the first half an hour or so, and then we'll resume the discussion for the next class. Okay. Uh, so just to update you. Okay. If you have any doubts throughout this entire process, because I'll be running by really fast through these topics, just interrupt and let me know, because we covered a lot of things, so I cannot go into that much detail. But yeah, I just want to brush up your memory on it. So we started off with our discussion on DNA, DNA structure, organization, and gene function, and different elements of DNA. So my pointers. Yeah. So we began with our discussion on discovery of DNA, where we talked about Frederick Miescher and how they identified that DNA is the starting material, or rather the material which carries all the genetic information which can be inherited across generations. Their first experiment, yeah, please keep in mind that you brush up on the experiments, and I will try not to include the scientist names everywhere because it's obviously not something very easy to recall. But for your own knowledge and for any future examinations you might be preparing for, if you're interested in research, it is very relevant. So please study it for your own good and your own career. But yeah, basically, this was the first experiment carried out by Misha, where he first saw that in leukocytes you could find something that was acidic in nature and had a basic portion, which were DNA and protein, which is something we have understood in a lot more detail in today's time. Then the next set of experiments, which were actually a lot more contributing and a lot more give a lot more insight, was carried out by Avery, MacLeod, and McCarthy, where they showed using experimental evidence that DNA is the bearer of genetic information. So they injected live encapsulated pathogenic bacteria into mice, and obviously saw that the mice died. And versus when they injected non-encapsulated bacteria, they were non-virulent, and therefore the mouse lived. When they mixed live pathogenic bacteria with heat-killed virulent bacteria, the mouse lived. But when they added the heat-killed virulent bacteria in which the DNA was intact with non-encapsulated bacteria, they saw that the mouse was dying. So it was actually the DNA component and not the capsule or the protein component present within the capsule that was contributing the pathogenicity of the virus, thereby suggesting that the virulence is encoded, or the information for virulence is encoded within the DNA sequence. So these were the scientists who actually made this quite remarkable discovery, and this was taken forward by Frederick Griffith as well. Then they extracted DNA from heat-killed pneumococci, removed the protein, and added it to non-virulent pneumococci, and that transformed the non-virulent or the non-pathogenic virus into pathogenic virus, and therefore the information was actually heritable. That could be concluded, and this was actually validated by another set of experiments known as the Hershey-Chase experiments, which were carried out by Alfred D. Hershey and Martha Chase, where they provided more evidence that DNA is actually the bearer of genetic information. So they radioactively labeled two different components of bacteriophage. One was the protein coat, the other was the genetic information carried in the DNA. And what they did was they used these labeled bacteriophage cultures and allowed them to infect into bacteria. And then they sort of agitated it into a blender, which removed the phage coat, and then they concentrated the cell pellet and then checked whether that cell pellet was radioactive or not. So in cases where the DNA actually had been radioactively labeled, which is what you see here. Sorry, what you see here, the 32P experiment, because phosphorus is a component of DNA. That part or those populations of cells which had been infected with the phospho-32 labeled bacteriophage actually were also radioactive themselves because the DNA which had been injected into them were radioactively labeled, versus when the protein coat was actually labeled using 35S, because sulfur is a part of a couple of amino acids like cysteine, etc., which forms the protein coat. In those cases, the coat was radioactive and the DNA which was injected was not. So from that information, they understood that the injected material, which is getting inherited also, is what is the genetic material. And so these were very simple experiments with very amazing conclusions that came out of them. Then the first study by the first structural study, which was published by Francis Crick and James Watson, uh, post analysis of Rosalind Franklin's data, was the extra diffraction strategy, which was used to decode the double helical structure of DNA. And Watson and Crick, they have been credited with this idea, along with Maurice Wilkins, who were awarded the Nobel Prize in 1962. So basically, this is what DNA is made up of. You have a sugar phosphate backbone, and this is the representation here. This is your sugar phosphate backbone, along with which you have these bases, nucleotide bases, which project towards the inside of the double helix. And yeah, so this is the templated polymerization of the new strand. And there are double bonds or triple bonds depending upon which bases are present. And among these also, we I think studied in a couple more slides as well. AT and GC are capable of hybridizing to each other, and their complementary base pairing persists. And yeah, so when there is polymerization, each of these nucleotide triphosphates keep on getting um, introduced, and the sugar phosphate backbone is built. And along with that, the sort of base pairing that exists between these bases is the hydrogen bonded base pair. And the DNA double helix looks something like this. Then we understood Chargaff's rules, which are very important in consideration or with respect to complementarity between DNA bases. So let's look over the rules one by one. Again, keep in mind these are also very question-friendly portions. So please make sure, and more importantly, you should know these things because these are basics. What I'm teaching you about the tools and the analysis, etc., all of that is something that you can refer to if you use any tool. If you go to its help section, if you go to its manual section, you will learn about, you can use those commands. In today's time, AI is very easily accessible. You can use that as well. I would strongly advise otherwise, but you have these tools at your disposal. But your basics, if they're not clear, that's going to affect your data analysis part, because that is something a tool cannot do for you. It's your own, you as a scientist, your brain is going to do that job. So please make sure your understanding is and your foundation is very clear regarding these things. So coming back to the discussion, Chargaff's rules included these following principles. First, that among different species, you would have variation in DNA-based composition. Again, this is something what I just mentioned as an answer to your question, that microbial genome can be segregated from human genome based on the arrangement. Of course, it's not the base composition that is going to affect it. It is actually the reference genome for microbes that has been made. But yeah, overall, that's just I wanted to connect these two ideas. Second, DNA specimens from different tissues of the same species have the same base composition. The composition does not change with an organism's age, state, or environment. And of course, A plus G equals T plus C. That is something we have already understood. And they were necessary to establish the 3D structure and of DNA and necessary to pass information from one generation to the next. Then came the Watson-Crick model or the structure of DNA. So they postulated these following points. First, there were two helical chains that were wound around the same axis to form a right-handed helix. There are other types of helices as well. I didn't go into all of that detail because that will become a molecular biology course. Maybe we can do that in the next semester, but yeah, you can read about it if it's interesting. There's an A form, B form, Z form of DNA. Very very rare types of DNA, and I will not include questions from things that I've not discussed in the class or that are not in the slide. But for your own curiosity, you can read about it. I think this was taken either from Leninger or from Voigt and Voigt, so you can refer to these books. You'll find all that information there. Then came the hydrophilic backbone of alternating deoxyribose and phosphate groups, which are towards the outside of the double helix, which faces the aqueous medium because they are hydrophilic in nature. The furanose ring of the deoxyribose, which is towards the inside of the helix, has these particular conformation known as the C2 prime endo conformation. Then you have purine and pyrimidine bases, which are stacked inside, and the base stacking is another force which stabilizes the double helical structure as well, and which makes them also perpendicular to the long axis. And there are two types of grooves which are formed, the major groove and the minor groove on the surface of the duplex. Why this is important is because this creates a site for the binding and docking of transcription factors, which will be able to recognize elements along the length of the sequence and then initiate transcription as and when necessary. So yeah, so that's why I was mentioning that understanding the basic structure is important because, like from here, we zoomed out a lot and we went into, binding of transcription factors. We zoomed out again. We went into the formation of nucleosomes. Then we zoomed out even further and we did high throughput sequencing and analysis. So we've gone back and forth a lot through this course. But don't worry, these are all independent topics that you can study as per your own comfort. Then we looked at some key definitions. For example, the gene, which is defined as a unit of hereditary information occupying a fixed position on a chromosome, which is called as a locus. So this is a very common schematic you'll find in any book related to epigenetics, which shows you the dimension of chromatin and DNA packaging. So chromatin is a double-stranded helical structure of DNA with approximately 2 nanometer width, which undergoes this sort of packaging and is wound around the histone protein octamer, which is made up of obviously 8 histone proteins around which DNA wraps 1.65 times. Why 1.65? They're because they are not two complete turns. Um, Basically, it's a mathematical calculation. It's just the dimension of the histone octamer and the length of the DNA, which is bound around the octamer, is not exactly divisible to result into its a fraction. So 1.65 turns come out of it. And apart from that, there is another histone known as histone H1, shown here in red, which sort of acts as a stopper, which limits the end of a chromosome, limits the end of a nucleosome, and it actually defines how far the default dynamic relaxation of the histone can actually occur. And I think I also discussed nucleosomal breathing somewhere when we were discussing epigenetics. But if not, nucleosomal breathing basically is a process where some amount of flexibility is allowed or some amount of dynamicity is allowed between the histone and DNA binding, whereby they are not, whereby they are allowed to relax by a couple of base, like around, I think it was around 20 to 50 bases, can get wound and unwound, and that does not require any additional chromatin remodeler or any energy investment. It simply occurs because of the dynamic nature of this binding. Then you have a chromatosome. The chromatosom actually defines this nucleosome plus H1 histone complex. The chromatosomes then form, they further undergo packaging to form the 30 nanometer fiber. And this arrangement usually occurs in a solenoidal fashion, following which there is further packaging, which forms these loops around 300 nanometer length that undergoes extensive packaging, finally forming the chromatid of a chromosome, which has a 250 nanometer individual fiber and approximately 1400 nanometer total dimension of a total width of a chromosome. So this is the amount of packaging it has undergone, that these meters and meters of DNA can be packaged into such a small portion. So that was the organization of the genome. And apart from the coding sequence, this extensive portion of the genome also has a number of different elements. What do they include? You have only around 1.5%, what you see here in red, this 1.5% is the protein coding part, which we are also, so interested and so bothered by. But a large portion is actually something that we usually do not take into consideration. But of course, there are multiple research groups who work in these domains. That includes line signs, transposons, unique sequences, introns, etc. And majorly, they are involved in regulating the expression and regulating the functionality of the coding part of the genome. So that is what we see here. Just give me a second. I want to switch. Yeah, I think it's working now. Okay. So, um, please let me know if at any point I'm not audible. I've just switched my mic. Okay. So, this is what the genome looks like. So, when we say what the genome looks like, it's basically an example of what chromosomes look like, and they have been stained. These are, of course, artificially colored. You do not get such a multicolored chromosome, but this actually shows you the difference in their dimensions, and also can answer a question you might not have immediately thought of, but why chromosome 22 was actually sequenced very early on, because it's among the tiniest chromosome. I think 21 is the smallest, but 22 was also pretty small, and it was the first chromosome that was sequenced in humans. And apart from that, you can also see that there's a sort of banding pattern in these chromosomes. You can read about it. It's something called as GMSR staining, which shows you different regions of the chromosome which stain differently because of the density of those regions. What density? You can read it up. Again, beyond the scope of this course, it is very core molecular cell and molecular biology, so we have not gone into details about that. Apart from that, this shows you the central dogma of molecular biology, where you have a DNA sequence which undergoes transcription to form mRNA and undergoes translation to form the protein, something we all know very well. And we also know that there are exceptions to this. There is reverse transcription, which occurs, which is capable of creating or which is capable of generating DNA from RNA, and it is something very commonly used in cDNA synthesis as well for measuring the expression of genes inside a cell or within a cell using qPCR. So there are exceptions to these rules also. Then we understood the structure of a gene and what are the different components. So if you look here, the most common region that we are interested in is this ORF, or the open reading frame, which obviously is composed of exons and introns. And apart from that, you have all of these regulatory elements which control the expression of that region. That includes the promoter which is shown here in red, and there are some control elements which are present very close to it, which are called the proximal elements of the promoter, proximal control elements for the promoter, and you have some distal elements which are much farther along. Now, when we say control elements, they can either be enhancers or they can be silencers. So they are going to be sensitive enough to allow the control over the expression of a gene depending upon what is the external stimulus to control that gene's expression. And these are altogether can be considered to be as upstream regulatory sequence. Similarly, you also have downstream regulatory sequences, which include some terminators and some distal control elements. Again, these can be enhancers or silencers, depending upon what protein binds to it and how its function can be controlled. And yeah, that's pretty much it. And also, the way in which these distal control elements usually work is that they can sometimes loop, because obviously DNA is a relatively flexible structure which has its own integrity, but its looping capacity is very important for gene function and gene regulation. So, when there is looping of this region over the promoter element, it can control the binding and the activity of RNA polymerase 2, which is usually responsible for active transcription. So that is what you see here. And here you can see human chromosome 22 in its mitotic conformation having two long double-stranded DNA molecules around 48 into 10 to the power 6 nucleotide base pairs long. Within this teeny tiny region, if you look here, you'll see approximately 10% of the arm has around 40 genes. And within those, 1% of the arm has 4 genes. For one gene, again, you have these exons, introns, regulatory sequences, etc., etc., etc. So that is the magnitude to which we are zooming in, and we are trying to figure out what is happening to DNA. Then what you see here are the different types of control elements and their mechanisms of working. So you have certain promoter elements, obviously, which are necessary for proper gene function. And it is possible that you might have some promoters which are going to favor transcription over others. They are called as strong promoters, and there are some which are weak promoters. Why it is important to have this knowledge also is because when you do restriction, digestion, or RDT, basically recombinant DNA technology, and you want to clone some sequence of your interest, and you wanted to have very high expression in your host system, you would prefer a strong promoter, obviously, over a weak promoter. And so usually what is chosen are viral promoters, because viruses have obviously have very active transcription of the genes of interest. So those are considered to be as strong promoters. Second, you might have enhancer sequences, and what enhancers also do, they can allow the recruitment of other transcription factors and other regulatory elements which enhance gene expression. Sometimes you may have boundary elements which help in looping, DNA looping, like I already mentioned, which brings some enhancer element close to the RNA pol2 bound promoter and allow transcriptional increase. So these are just some common ways in which these functions can get enhanced. And then we talked about regulating gene expression or gene function. Commonly, there are six steps at which this can be done. First is in the conversion or the transcription of DNA to RNA. There's transcriptional control. Next, there is post-transcriptional control or RNA processing control, which is somewhere. So here you see from DNA to RNA transcript, you have controlled it. Second, from RNA transcript to mRNA, where it gets capped and tailed and is now destined to be an expressed RNA or an mRNA, messenger RNA. And after that, after translocating from the nucleus into the cytosol, you can have RNA transport and localization control. This is another checkpoint. Then the mRNA can either be subjected or be cleared for translational control, or it can be impeded from doing so. Then you have mRNA degradation control. So any inactive mRNA can be generated from that. Then the encoded protein can also have control over the activity, where its active or inactive state can also be controlled. So these are many different checkpoints which can be controlled externally or by the cell itself depending upon the condition and upon its sensitivity to different stimuli. Then we saw the typical gene control region for a eukaryotic gene. So it's similar to what I've already discussed. So very close to your actual gene of interest, you have these promoter sequences to which RNA-pol2 binds, and this binding is dependent upon general transcription factors which are present. Then there is different components of the RNA-pol2 are also responsible for it, the sigma unit and so on and so forth. Then you have co-activators, which can be very close to the promoter, which can be quite far. You have these cis regulatory sequences, which when bound by trans factors can act as activators or co-activators of the transcriptional process. And the reverse is also possible, where it can impede the expression of a gene as well. So depending on this, the gene function can be controlled. And here what you see is pretty much an example of the looping I was talking about. And you can see that to a same scaffold protein, this sort of lilac purple colored protein, to that you have different elements binding, which are all together forming a complex that is going to actively promote transcription for gene X at a very high rate. So this is sometimes called as a mediator or a scaffold protein, which facilitates the process of transcription and selectively enhances or empowers it. Then you have different strategies by which eukaryotic repressors can work. So you can either have competitive DNA binding. So activator proteins and repressors can sometimes compete for the same regulatory sequence. You have masking of activation surface, where the binding site for repressor and binding site for activator are both bound by the respective proteins, but the repressor binds to the activator's main domain and prevents it from doing its job, prevents it from activating. Then the repressor can block assembly of GTFs or general transcription factors. Like you see here, the assembly of the transcription factor has occurred, but activator or an enhancer is not able to access it because repressor has already bound and sort of immobilized it, sequestered it. Then you have the recruitment of chromatin remodeling complexes, um, which are obviously going to control how tightly or how loosely a certain region of the genome can be exposed. And depending on that, the transcription from that site may be taken forward or not. Then you have recruitment of histone deacetylases, which are going to remove acetyl groups, or there can be recruitment of histone methyltransferases, which will which will which will put a sort of a repressive mark, a repressive mark, and maintain the transcriptional silencing of a certain region of the genome. So these are examples of different ways in which gene function, silencing, and activation can take place. Then you have this schematic that summarizes properties of insulators and barrier sequences. So as you can see, there's a domain of actively transcribed chromatin that includes this gene B within that sequence itself. And you have insulator elements shown here in blue on either side of gene B. So depending on whether or not these sites are bound by an insulator binding protein, which is shown here in figure B, transcription may or may not be allowed to proceed. So yeah, insulator binding protein sort of loops out the chromatin and favors and it favors correct cis-regulatory sequence and gene associations, and gene B gets regulated and they cannot influence the transcription of gene A. So you're creating a local site where transcription can be actively initiated. Um, yeah. Then you have multiple inputs at the same promoter, so multiple sets of transcription regulators, coactivators, co-repressors, all of these can work together to influence transcriptional initiation at a promoter. And it is likely that final transcription results from a sort of a competition or the sum. Sorry, this issue. Yeah. results from a competition between activators and repressors. So there is a summation of all of the different sort of stimuli which are being sent to the cell, based on which finally the decision is made by the cell to either proceed or not proceed for transcription. So that was the discussion we did in week one, and we covered a lot of different topics. So again, any doubts, please interrupt me. We can have that discussion, and then we'll move forward. Please let me know if there's anything you want cleared. Okay. Uh, yeah. Next, in week two, we had moved on to discussions about the role of DNA methylation in gene expression. So it is an epigenetic modification of DNA. Just a second. Yeah, then we moved on to mutation. Okay, so it's an epigenetic modification of DNA that's important for normal control of transcription, for embryonic development, genomic imprinting, etc. It is controlled by a set of enzymes called as DNA methyltransferases or DNMTs, methyl CpG binding proteins, and other chromatin remodeling factors. So commonly in cancer, DNA methylation patterns can be very extensively disrupted, and genome-wide hypomethylation or gene-specific hypermethylation events can occur within the same cell. So the control can be very different. Many different diseases are also often associated with these improper epigenetic marks that are necessary for normal development and sustenance of cells. Just a second, I'm getting a call. Yeah. So yeah, I was talking about the different diseases that are associated with improper or loss of normal imprinting, including the Prader-Willi syndrome, some neonatal diabetes, Angelman syndrome, etc. And then we talked a little bit about DNA methylation and cancer and how different ways in which these modifications can affect it. So two possibilities, like I've already mentioned, either there can be hypomethylation of CpG islands, which will lead to transcriptional repression and loss of tumor suppressor expression. When there is hypermethylation of CpG islands, it can lead to silencing or suppression of tumor suppressor genes, which can aid the progression or the onset of cancer. The other possibility is that there is hypermethylation. Normally, there is hypermethylation of heterochromatin regions. If there is some sort of genomic instability, there is recombination of those silent sites, and they suddenly start expressing. One example of which are oncogenes, which are often present within these regions. Then that can also lead to cancer. So this was just one example, but of course there are different possibilities as well. And this was the model that showed how normal DNA methylation can be disrupted in cancer cells. So you have this normal versus tumor cell. In normal cell, you expect hypomethylation of the tumor suppressor and hypermethylation of oncogenes. And there can be some shift or some imbalance in this methylation status, which can eventually lead to development of cancer. And this was a detailed example of what can happen in a normal and a cancer cell. So you can have hypermethylated repetitive DNA in a normal cell, which is sort of not expressed at a very high level, but there can be some shift in this. If there is hypomethylation, then it can result in genomic instability at those sites, and that can generate double-stranded RNAs, which can bring the repressive histone modification machinery, DNMTs, MBDs, etc., which can also silence the tumor suppressor gene. Basically, what we understood from here is that there can be loss of methylation marks in some regions of the genome, and there can be also gain in methylation of some maintenance genes like TP53 or any other tumor suppressor, which can upset this balance between oncogenes and tumor suppressors and eventually lead to the progression of diseases as deadly as cancer. Yeah, despite the near perfect fidelity of genetic replication, infrequent unrepaired mistakes in the replication process can lead to changes producing something we understand very well as mutations. There are two possibilities or two ways in which they can arise. Some of them can be spontaneous, some of them can be mutagen induced. So mutagen induced, of course, is reliant on the presence of some sort of an external mutagen, whether that is chemical, whether that is ionizing radiation, etc. And spontaneous errors are usually classified under the group of DNA polymerase slippage or DNA poly associated errors, which affects the ability of or which basically changes the sequence at that region and gets passed on over multiple generations. So there are, of course, repair machineries which are present there, but if there is if an error goes unrecognized, then that can get incorporated into the genome, and if sustained, that can potentially lead to a genetic mutation. So this shows the exact effect that I just mentioned. You can go through it once more. Then we talked about the genetic code. Um, there were five key properties of the genetic code. First, that it is degenerate. Most amino acids can be specified by more than one codon. They are non-overlapping, so codons are read in a continuous non-overlapping manner. So three at a time, three at a time, three at a time. That's how it's read. There's not an there are no overlapping frames. It is commaless. There are no punctuation marks between the codons. They are all read one after the other. Then they are non-ambiguous, which means that one codon only specifies one amino acid. They're universal. It is conserved in nearly all living organisms. Then we came to the idea why we discussed codons are because there are different sort of mutations which can change the fidelity or the original codon which was being encoded. So you have different sets of mutations. The first set we will be discussing. Just let me quickly check the time. Okay, yeah, we'll wrap up the class in another maximum 15 minutes. So whatever portions can be covered in that duration, we'll take those up. So under point mutations, you have a change in a single nucleotide base that would be recognized as one single base mutation or a point mutation, which can be further classified into either a silent, missense, or nonsense mutation. What is silent mutation? Once um, basically sequence mutations where you do not have an altered protein sequence or an altered amino acid sequence, that is a silent mutation. Example shown here basically shows you valine. In both cases, valine is getting encoded. One has GTA, the other has GTT. Why this is feasible is because of the degeneracy of the genetic code, which is why the same amino acid gets encoded and the mutation effectively gets silenced. The other possibility where the mutation is not silenced and the amino acid is actually altered would be called as a missense mutation. So what do you see here? You see that there is a change from proline to threonine and nonsense, similar to missense mutation. The only difference being that instead of a coding amino acid, you now have a stop codon. So these are the three major types. You can again brush up on this. So what would be the outcome of this particular sort of mutation? The nonsense mutation is going to lead to a truncated protein or an incomplete protein, which is of course not probably not going to be functional and can actually be disastrous as well. So that is what we saw. Then we saw the different, we saw this in more depth. So like you can see here, this is the premature truncated protein that is getting synthesized from the ribosome. Why so? Because the change in the amino acid sequence has, instead of recruiting the next charged tRNA, it is recruiting a release factor, which is going to terminate the which is going to lead to the altered codon, which is now encoding for a nonsense codon that is going to recruit the release factor that is going to release our polypeptide, and an incomplete or truncated polypeptide chain is going to be synthesized. Here we saw how a mutation can get incorporated across multiple replication cycles. So from your parental DNA, if at the time of DNA replication, one single error is made by the polymerase sequence, then every time that base pair or every time that DNA undergoes replication at the fork, that
first generation to second generation progeny, the mutant population would be generated. How so? You have GC here in the original parental DNA. If by chance at the time of DNA replication, DNA polymerase has committed an error or committed a mistake, and it has incorporated a T or a thymine opposite a guanine. While there will be a single kink initially, if it goes unrecognized, then in the progeny, what would you find? You'd find a GT and a GC in the first generation. When this is taken forward, what happens in the next generation is that the G and T again separate at the replication fork, and then across G you will have C incorporated, and across T you would have A incorporated. Now this base pairing is going to be stable, and this has made a permanent change in the sequence of the DNA in that region and can now be recognized as mutant. So what you see is that the first generation progeny might or might not have, it is possible that in the first generation progeny, the repaired machinery is able to recognize it and is able to fix the replication associated error. Majorly, that is carried out by the MMR or mismatch repair machinery. But if it does not, then in the second generation, it becomes a permanent mutation, making it unrecognizable, irreparable, and in some cases, some extreme cases, even disease causing. So therefore, polymerase slippage is a major problem. Also, it is also one source of diversity that helps evolution. But of course, if it occurs within some functional protein, then that can become more problematic than beneficial. The next set of mutations are the frame shift mutations, which literally, as the name suggests, results in the shift of a frame, which is being used by the ribosome to read the mRNA sequence. So, insertions or nucleotide deletions can result in a shift of the reading frame or the insertion of a stop codon. So, what you see here is that there can be a change in the frame. Let's look at this example. So you have the unmutated original mRNA sequence. If there is a single nucleotide insertion here between C and G, if there's a single nucleotide insertion, what it does is that between C and C, it is inserting a G nucleotide, then it is going to completely shift the frame, as a result of which now you will have completely different amino acids encoded right after methionine. If there's a double nucleotide insertion, then that is going to lead to another shift in frame. However, if there is a triple nucleotide insertion, what it does is it retains the codon frame, but it accidentally incorporates a new amino acid at an unexpected location. So the protein possibly may be functional if there are three base pair insertions, but you cannot always know that. It may or may not be functional, but it will certainly have a new amino acid incorporated and at an unexpected location. Now, why can we not be very certain that the protein function may or may not work? Sorry. So it actually depends upon the nature of the amino acid. Say, for example, if it is a charged amino acid that is getting incorporated, like for example, here if you have glutamic acid or you have histidine, these kind of amino acids, then they are capable of forming new sorts of interactions. They are capable of affecting the chemistry of the protein, because of which it may not fold properly. It may not hold the secondary structure or the shape or the domain organization which is necessary for the protein's function, or it may change the nature or change the capacity of a protein to release a certain ligand. A number of possibilities exist, but basically what it does is that it retains the overall polypeptide sequence except for the insertion of one single amino acid. So this is an example of frame shift mutations. And yeah, in-frame mutations can occur where an amino acid may be added or lost that can potentially affect protein function. And overall, this was a recap of all of the different types of mutations that can exist. There are transitions and transversion mutations. Transition mutation leads to an AT to GC switch. Transversion mutation leads to a CG to GC switch, where the nature of the purines and pyrimidines are maintained, but it leads to a switch in the sense and antisense. As a result, the overall frame will be shifted. For example, if you look here, if you have TCT and then you have CAA, The transversion mutation is going to lead to TCT and GAA. So the amino acids which would be encoded in those under those circumstances would be completely different. Then you have missense mutation, like I already discussed, where there's a change from one amino acid to another. Here you can see a shift from lysine to glutamic acid. There can be a nonsense mutation, which leads to truncated protein generation or translation of a truncated protein, which is going to lead to a lysine to stop codon conversion, which is going to terminate the protein at an unexpected site. Then you have a neutral mutation where there's a change from one amino acid to another, where the property or the chemical nature of the amino acid is intact. So here you have a lysine to an arginine switch, which is going to be a relatively quieter mutation. The mutation is there, its effect possibly will be seen, but the disruption that the mutation is going to cause is going to be slightly lesser because the nature of or the positive charge of the amino acid is staying conserved. Then you have silent mutations, such that silent mutations are those where the same amino acid is getting specified, like we saw our previous example from valine to valine. You saw the same amino acid get encoded. So here you see lysine and lysine in both circumstances. So the nature or the overall organization will not get disrupted because of the degeneracy of the genetic code. And then you have frame shift mutations, which are going to lead to a complete change in the amino acid sequence, depending upon whether there is a single base, two base, or three base insertion which is occurring there. Um, then we talked about how these mutations arise. The first set, like I already have discussed, it is polymerase errors, which can lead to this possibility where there's initially a single base pair error, but if that is allowed to pass on, then it can lead to looping out of the template strand and it can lead to second generation mutant generation. And the second possibility is because of second possibility is because of chemical mutagens. Here, what you see are an example of different base states, which are called as tautomers. So tautomers are different chemical states of the same nucleotide bases. So basically in DNA, the keto form of each base is usually present in the double helix and is responsible for normal Watson-Crick base pairing, where you see AT and you see CG base pairing between these DNTPs or nucleotide triphosphates or nucleotide. Uh, for Watson-Crick base pairing, what can sometimes happen is that if there's a switch from the normal keto form of guanine to the rare enol form of guanine, then guanine can start forming base pairs with thymine. And for adenine as well, you can see mostly it is seen for adenine and guanine only. So you can see that the rare imino form of adenine is able to form base pair with C. So you have a GT and AC base pair under those circumstances, which is of course not normal, but it is an error and it will be recognized as a mutation. So that is another possibility. Spontaneous chemical changes include depurination and deamination also of certain bases, which create lesions. So, for example, cytosine, when it undergoes deamination, but their amine group is gotten rid of, you will find the generation of uracil, which is a major source of error, very commonly found as well. And it is the removal of an amino group from a base. And normally it is not expected in DNA, but this can occur under a large number of circumstances. When it does happen, uracil under most circumstances is recognized by base excision repair machinery or the BER machinery, and then it removes uracil. And it basically what it does is there's an enzyme called uracil D-glycosylase, if I'm not wrong, UDG. Yeah, I think that's the name of the enzyme, which can detect uracil in the genome and it removes that site. And in the next round of replication, that site again gets filled up based on what is the not even during replication. BER itself is also able to incorporate a base there, but it is basically responsible for removing uracil. The other possibility is depurination, where there can be loss of purine from DNA, generating an apurinic site. And there are also ionizing radiations like UV light, which can increase the chemical energy of certain molecules like pyrimidines, and it can form pyrimidine-pyrimidine dimers. most commonly are thymine dimers, which are, as you can see, structurally very large entities which create a sort of hindrance to the polymerase machinery, and polymerase can get stuck during replication, and therefore they pose a major problem to the replication machinery. So these are also recognized and also removed by repair machineries. But yes, the main problem is that sometimes detection is not possible. And secondly, even if detection is possible, sometimes some cells they accommodate so many errors that their replication machinery is not able to cope up with the magnitude of mutations and magnitude of structural aberrations, that some mutations and some aberrations are allowed to persist, and then that can become very lethal later on. So then there are chemical mutagens, like there are base analogs, which an example of which is 5-bromouracil, which, if incorporated in its normal state, pairs with adenine. If it changes into its rare state, it can base pair with guanine, which is what you see here in these examples also. So 5-Bu behaves like thymine, binds to adenine, but under rare circumstances in a rare state, it can behave like cytosine and form base pairs with guanine, and therefore it leads to a switch from it. It can induce a CG to a TA transition as well. You can go through this example to see how that mutation can become persistent if it is allowed to persist for a if 5-bromouracil is present in the medium for a sufficiently long time. Then you have intercalating agents like proflavin, acridin, ETBR, ethidium bromide, etc., which intercalate or insert themselves between adjacent bases of the DNA double helix, and therefore they can create mutations. So when a replication occurs, then that region doesn't actually have a base, and so any random base can get inserted at the time of replication, and then that is going to create errors, and that error will stay or will persist and will eventually be forming a mutation. So yeah, I think we'll take a pause here. We discussed the first 2 weeks. Rest you can refer to slides and recordings. All every material that has been covered till this time, I think, has been already discussed. I'll still tell you till what portion you should brush up. Just let me. Um, I'm thinking we'll skip modification epigenetics for now. Maybe we'll keep it in the next test. Otherwise, it's going to be a lot of material to brush up on. We will keep it till yeah, Sanger sequencing. Yeah, so we'll cover these topics: the basics of molecular biology, gene structure, function regulation, mutations, polymorphisms, and introduction to sequencing methods, Sanger. We'll keep it till here so you can read up and quickly brush up on these, and then we'll have a test, and then we'll proceed with our discussions for the next class. Any doubts, anything you want clarified before we take a pause for today?
No,
Nothing?
ma'am,
All good?
still here. It is clear. Just we have to
Oh,
revise.
yeah. These were relatively easy topics which you probably have studied before, so I think it should be okay only. But yeah, I'll drop an email, and we'll meet on Tuesday next week. Okay, so thank you for joining. I'll see you again next Tuesday. Have a good night. Thank you. Bye.
Thank you, ma'am.
Thank you, ma'am. Ma'am, can I ask some more question by
Yeah, yeah,
emailing you?
sure.
That
No problem. You can drop an email.
because
No
right
problem.
now I don't recall any question.
Okay, okay, yeah, no problem. You can ask me. No problem.
Thank you, ma'am.
Okay, bye. Good night.